So today I'm going to talk about one of our recent papers that was just published from some research that came out of my lab. So this is a paper that was just published in the journal Photochemistry and Photobiology. And this is the master's thesis work of a graduate student in my lab, Abdul Rahman. So the title of this paper is Insulin-like growth factor 1 impacts p53 target gene induction in UV-irradiated keratinocytes in human skin. And so sort of background information on this topic is we know that non-melanoma skin cancers, for example, are a big and growing problem, and they're caused by UV wavelengths of light that cause mutations. However, there are other factors that likely impact non-melanoma skin cancer risk, and we're interested in understanding what some of those factors are. And what I'll try to convince you today, and this paper focuses on, is a role for a hormone known as insulin-like growth factor. So we know that IGF-1 is produced by fibroblasts within the skin, in human skin. And as people age, we know the IGF-1 levels decrease in abundance. And what we think happens is that as people age, there's more senescent fibroblasts. This leads to reduced production of IGF-1, and therefore keratinocytes in the epidermis have less IGF-1 in the environment, and that may affect how they respond to things like UV light. Now, previous work by us and others have shown that there is a difference in how keratinocytes respond to things like DNA damage if they're grown in the absence of UV. So previous studies have looked at DNA repair, ATR kinase signaling, which regulates DNA synthesis. So the purpose of this paper is to look at for a role of p53 in the response of keratinocytes to UV. So we know that p53 is a tumor suppressor gene, is known to become activated after UV, where it acts as a transcription factor to regulate the expression of a variety of different gene products. Some of this we'll look at in this paper. One of which is p21, which is a protein that is known to inhibit cyclin-dependent kinases, they're involved in promoting cell cycle progression. And thus, when UVB induces p53 to act as a transcription factor to upregulate p21, the thought is there's more p21, less cell cycle progression. However, p53 also regulates genes involved in nucleotide excision repair, including XPC and TDP2. And also, somewhat less studied is POL-eta, which is something involved in translucent synthesis. So POL-eta is a specialized DNA polymerase that can actually allow efficient DNA synthesis across UV photoproducts, whereas most polymerases are unable to replicate across that. And POL-eta is actually mutated in a variant form of xeroderma pigmentosum, disease that leads to high rates of skin cancer. So although p53 has been looked at in the regulation of several of these gene products previously, it really has not been looked at in the context of human skin. That's what we wanted to do here, and specifically how deregulation of IGF-1 affects this response. So in figure 1b, what we did was to actually take telomerase immortalized keratinocytes grown in culture, grow them for 24 hours in the absence or presence of IGF-1. Again, it's just a small growth factor that we can add to the cell culture medium. And then we expose cells to UV, in this case, 100 joules per square meter of UVB. And then at the indicated time point, cells were harvested for Western blotting to look at the phosphorylation of p53. This phosphorylation is a good marker for the activation of p53, since the phosphorylation helps to stabilize the protein. Normally, p53 has a short half-life. And so we can see it within 30 minutes, for example, after UV, we begin to see more p53 phosphorylation. However, in cells grown in the absence of IGF-1, we still see this induction of phosphorylation, but it's reduced, indicating that p53 is not being properly activated in these cells after UV exposure when IGF-1 is missing from the cell culture media. So what are the consequences of that? Again, based on the figure up here, we can look at the induction of these gene products. And so in figure 1c, we did this by Western blotting, looking at the protein levels. And we can see very nicely that several of these gene products are indeed induced by UVB, and this is at a 24-hour time point after UV exposure. So we can see, for example, POL-eta increases in abundance after UV, when cells are grown with IGF-1. Similar result with P21, somewhat less so with XPC, and almost no response with DDB2, indicating that there may be differences in keratinocytes in comparison to other cell types with regards to the induction of these gene products. However, again, if cells are grown without IGF-1, now we fail to see induction of any of these gene products. So it looks like we need IGF-1 for p53 to properly function and to turn on the expression of these important gene products. In figure 1D, we basically essentially quantify the relative protein induction. So looking at the protein levels with UV to no UV in cells grown even in the presence or absence of IGF-1, and looking at these different products. So just essentially a quantification of the results in figure 1C from several different independent experiments. Each dot here represents an individual experiment showing, again, there's somewhat variability from experiment to experiment, but on average, the genes are induced to a significantly greater extent in the presence of IGF-1 than in the absence of IGF-1. So this is looking at things at the protein level. We know that these p53 is a transcription factor that increases RNA expression. We see very similar results when we look at the RNA levels of pol-eta and p21. So it correlates reasonably well with the protein data. So in the second figure of the paper, we wanted to look at this further, look at more time points and different UV doses, since figure 1 was essentially 24 hours after 100 joules UVB. So in figure 2A, we do a dose titration, looking at increasing UV doses, and we see a nice dose-dependent increase in pol-eta and p21. The cells grown in the presence of IGF-1, but in the absence of IGF-1, that induction is significantly reduced. And in figure 1 or 2B and C, we quantify those data from several independent experiments. In figure 2C, what we're looking at is the time course. This is after a single dose of 100 joules per square meter UVB. So again, the protein seems to be most highly increased at between 24 and 48 hours after UV exposure. So it looks like it's a reasonably stable induction. However, in cells grown in the absence of IGF-1, there really is very little induction after UV exposure. Now, in figure 2D, we also want to look at what the effect of multiple exposures of UV. So in reality, when we go out in the sun, we're exposed to the sun every day. So we're constantly exposed to UV. We wanted to somewhat mimic that in a very simplified system. So what we did was to take these telomerase immortalized keratinocytes grown in the presence or absence of IGF-1, and then on 2 subsequent days expose the cells to 100 joules per square meter UVB. And so just like we showed previously, we see the phosphorylation of p53 is induced under these conditions. p21 and pol-eta are induced. However, in cells grown in the absence of IGF-1, that level is significantly abrogated by the loss of IGF-1. Now another way of mimicking this response is to actually actively inhibit the IGF-1 receptor, since IGF-1 binds to the IGF-1 receptor on the cell surface, and then that can mediate other events inside the cell. So to mimic that, we can also use a small molecule inhibitor. So we use an IGF-1 receptor inhibitor known as AG538, treated at 10 micromolar for 30 minutes, and then expose cells to UV. We see a very similar response. So cells treated with the IGF-1 receptor inhibitor failed to induce pol-eta and p21 to the same extent as cells treated with a vehicle control. Now, to be sure this is dependent upon p53, we used a small molecule p53 inhibitor. See a very similar thing. Obviously, that in the absence of active p53, these genes fail to be induced. So what are the functional consequences of this loss of induction? Now, if we look, we might predict based on the failure to induce p21 that cells might preferentially move into S phase. However, when we used pyrimidine staining to look at cell cycle distribution in the absence or presence of UV, again grown in the absence or presence of IGF-1, we really didn't see any significant difference in rough cell cycle distribution, suggesting that, at least under these conditions with these doses, there's not a significant effect on cell cycle distribution. We also did an assay to measure the amount of DNA synthesis taking place in the cells, or the number of cells that are replicating, by incorporating a nucleotide analog known as EDU into these cells, which can then be visualized through chemical means by immunofluorescence microscopy. And both in the presence and absence of IGF-1, we actually see that UV seems to induce more proliferation. And there's evidence in the literature that UV may actually induce stimulation or proliferation. Although there's a slight trend, a little bit higher levels of ED positive cells in the absence of IGF-1 was not statistically significant from the cells grown in the presence of IGF-1. We also looked at cell survival by two different means. One is through what we know is called an MTT assay, looking at essentially acute cell survival a few days after UV exposure. And there really is not significant difference. There's, again, a slight trend that in the absence of IGF-1, there's a little bit less survival, but it was not statistically significant compared to cells grown in the presence of IGF-1. However, we also did what's known as a clonogenic survival assay, where cells are plated, essentially a low number. There are single cells on a plate. Look for the ability of those cells to divide and form colonies. We can see in both treatment groups, both in the absence and presence of IGF-1, there's a reduction in the number of cells that can form colonies in the presence of UV, or cells that were exposed to UV. However, there was a much more significant difference in cells grown in the absence of IGF-1, and only during that initial UV exposure. To get colonies to actually grow, you have to have IGF-1 in the medium. So it looks like that the absence of IGF-1 in the media does lead to potentially a long-term consequence in the clonogenic survival of these cells. So everything we've shown so far is looking at keratinocytes in vitro, and that's all valuable and useful. It's a nice experimental system. We next wanted to look at this in the context of actual human skin. And so what the system we use is actually use discarded skin from routine abdominal plasty surgeries. There's ways we can actually inhibit IGF-1. But one of the things we've done over the course of the last 4 years is actually collect samples from a number of different people of different ages. And so in figure 5A, what we've looked at is do quantitative PCR to look at the mRNA levels of IGF-1, again, relative to an internal controlled beta microglobulin in people from different ages. And so we can see here that there's quite some variability. These are human subjects, essentially, some variability in the level of IGF-1. But we do see a trend that as people age from their 30s to the mid-60s, there is a decrease in the level of IGF-1 in the skin. So now to then mimic this in an experimental system, we can actually use this IGF-1 receptor inhibitor I mentioned earlier, AG538. We essentially topically apply to the skin and then expose the skin to UV. And then at 24 hours later, we can harvest the skin, harvest the epidermis, and look at the RNA levels of the genes we've mentioned, POL-eta and P21. So we can see that both genes seem to be induced by UV. So again, this is looking at the relative RNA, the plus UV to no UV sample, skin specimen. In the case of POL-eta, there's about a 2.5-fold or so on average increase in POL-eta levels. P21 is induced a little bit higher levels, about 4-fold. However, when we topically apply the IGF-1 receptor inhibitor, it seems to block this induction. So it's significantly reduced, or very little induction of these genes at the RNA level. And we also want to validate that at the protein level. And so in figure C, we've done Western blotting, either 1 hour to look at p53 phosphorylation, which occurs quickly after UV exposure, and see that the IGF-1 receptor inhibitor blocks that phosphorylation induction. And then we also looked at the protein levels of P21. So we can see very nicely that P21 levels increase dramatically in the UV treated skin. However, when the skin is treated with the IGF-1 receptor inhibitor, that stimulation induction is abrogated. We've tried to look at POL-eta, but at least with the antibodies we have on hand thus far that are commercially available, they don't work well in the human skin samples. And in figure 1D, we've actually looked at this induction. So again, it's about a 4 to 5-fold increase on average of POL-eta level, of P21 levels, after UV exposure. But if the IGF-1 signaling is abrogated by topically applying AG538, that induction is significantly reduced or is completely gone. So these results suggest that in human skin and in keratinocytes in vitro, that IGF-1 plays an important role in the ability of p53 to induce these different gene products. And thus, based on evidence that IGF-1 expression is decreased in geriatric skin, we hypothesize that if we were to look at the level of these genes and proteins in geriatric skin, they may be lower than in young adult skin. And this is one of the further studies we're working on in the future. Moreover, we have some indication, because POL-eta is involved in in this process of translesion synthesis, that the inability to induce this polymerase might mean there may be other polymerases that are more mutagenic, less able to accurately replicate across thymine dimers induced by UV, and therefore might lead to more mutations and may be responsible for skin cancers. And therefore, geriatric skin, because of a failure to induce B21 and POL-eta, may result in more mutagenesis. And with that, thank you for your attention.